Genes, or sequences of DNA and RNA, encode proteins, and proteins dictate cell function. Although DNA is comprised of only 4 building blocks called A, T, C, and G, their unique sequences produce tens of thousands of different proteins. The combination of these few letters essentially makes up every living thing. However, mistakes within the DNA sequence can occur. Mutations, which can be inherited or spontaneous, can damage or change the DNA so that the genetic message carried by a gene is altered. In other words, any changes to DNA can potentially cause abnormalities in the structures of the resulting proteins and, therefore, in their functions. Since proteins are responsible for many life processes, a protein with altered function due to a mutation can lead to disease. Given that protein impairments result from genetic defects in the form of mutations, it has been a major initiative of scientific research to map out which genes correspond to the production and regulation of specific proteins. This research came to be known as the Human Genome Project, a large effort in which scientists successfully sequenced the complete human genome and mapped over 20,000 genes found in the human body. Although the project was completed over a decade ago, the Human Genome Project should not be viewed as an end in itself. Rather, it marks the start of an exciting new era of the genome in medicine and health. This massive effort has paid off by providing us with a better understanding of how abnormal genes cause disease. The focus at Intellimedix is to treat genetic diseases by determining and characterizing the underlying mutations in a person's genome, and then to identify existing drugs that can treat the diseases caused by those mutations. By sequencing someone's genome, we decipher the genetic code for that person's proteins and can determine which proteins are altered. By investigating exactly how the underlying mutations alter specific proteins and lead to disorders, we can find the most effective drugs to treat any diseases caused by those mutations. We accomplish this by employing our advanced computational and data mining algorithms to create a database of drugs and their binding protein targets. Since dysfunctional proteins can be the cause of disease, drugs aimed at fixing the protein's effects are ultimately expected to treat the disease. Coupled with available online databases, our novel computational approaches allow us to further personalize an individual's medical treatment. During our drug screening process, we focus on previously FDA-approved compounds to find alternative uses for them. Our technology predicts new applications for thousands of FDA-approved drugs based on experimental drug-protein interactions. However, drugs come with side effects, and the occurrence of adverse side effects in patients depends on how they react to the molecular compounds. Keeping this in mind, the proprietary technology developed at Intellimedix provides a more complete picture of a given chemical compound and how it interacts with specific genes, including its protein targets, side effects, and toxicity. This comprehensive understanding helps us suggest the most effective compound to treat a patient's genetic disease in a less expensive and accelerated manner than traditional diagnostic methods. Through a thorough genetic analysis, Intellimedix's scientists can recommend the most effective treatment options to doctors for their patients in an individualized and personal manner. This process encapsulates the idea behind personalized medicine. We can tailor disease treatment to the individual since the medications being prescribed are targeted to the specific mutations that an individual has. By using a highly integrated approach, Intellimedix can offer better clinical interpretations and treatments for genetic diseases.